Hi viewers, today we are going to discuss the structure and function of the nephron. Under this topic, we are going to discuss the structure of mammal kidney, usually kidney of human beings, structure of nephron, apart from this, Malphigian corpuscle, Malphigian body, and the renal tubule, and the types of nephrons like the cortical nephrons, juxtamedullary nephrons. So let us see one by one. Structure of the kidney. One pair of kidneys are present in the human being. They are bean shaped. One pair of kidneys are present which are bean shaped, dark red in color, and they are metanephric kidneys. As we know, protonephric kidneys are very important in the competitive examination point of view. Protonephric kidneys are present in the hagfish or mixin. Mesonephric kidneys are present in the petromyzon, fishes, and amphibians, whereas metanephric kidneys are present in the reptiles, aves, and the mammals. We should remember this. So, one pair of bean shaped, dark red colored metanephric kidneys are present. Then coming to the location, where actually they are located in the body of the human beings. They are located on either sides of the vertebral column, in the abdominal region, between the dorsal abdominal wall and the parietal peritoneum. Between the dorsal abdominal wall and the parietal peritoneum. And one more important thing we have to discuss here, it is these kidneys are known as the retroperitoneal organs. Retroperitoneal organs. Because if you observe the heart, it is covered by the pericardium, and if you observe the lungs, pleuron membrane, right? At the same time, the kidneys also covered by the peritoneum membrane, but this peritoneal membrane covers the kidneys in the ventral side only. Ventral side of the kidneys covered by the peritoneal membrane, and the remaining sides that will be empty without any peritoneal. So that it is known as the retroperitoneal organs. These are the one and only organs which are retroperitoneal in nature. Retroperitoneal in nature. Kidneys generally called as the retroperitoneal organs, that is, they are covered by peritoneum on ventral side only. Right? Then, each kidney is ensheathed by a fibrous renal capsule, and the capsule is again surrounded by the perirenal fat. Right? And if you observe the arrangement of the kidneys, see here, if you draw a line, this is the left kidney and this is the right kidney. Left kidney is somewhat anterior than the right kidney. It is somewhat behind. Left kidney is arranged anteriorly and right kidney somewhat behind the left kidney it is arranged. This is because if you observe the digestive system of the human beings, in the abdominal cavity, right abdominal cavity, there will be a presence of liver. Due to the presence of the liver, it pushes back the kidney, so that the right side kidney is somewhat arranged backward. Okay, that should be remembered. And then kidney on the outer side it is convex, whereas inner side, that is towards the vertebral column, that is concave in nature. outer side is convex and inner side is concave. And at the concave side, we have a notch-like structure. We have a notch-like structure which is called as the hilum. Which is called as the hilum. That hilum's opening into the kidney, opening of the hilum is known as the hilus. Opening of the hilum is known as the hilus. At the same notch, we will see the entry of the red color one is the artery, blue color one is the vein. So here there is a entry of renal artery inside to the renal artery inside to the kidney. Actually it is formed from, so here also you can see, so each kidney is supplied with a renal artery. Actually it is formed from the dorsal aorta, a branch from the dorsal aorta, and it supplies oxygen and nitrogenous wastage for the filtration to the kidney. It supplies oxygen and the nitrogen wastages to the kidney. And just beside the renal artery, we will also see a blue color line, that is the renal vein. It is known as the renal vein. Renal vein collects the impure blood, that is, it collects the carbon dioxide which is released in the kidney, and it is supplies this carbon dioxide blood, deoxygenated blood, to that of the posterior vena cava. Okay, it collects the carbon dioxide blood and mixed up with the large vein, renal vein, and goes and mixed up with the posterior vena cava. Posterior vena cava. Right, that is the supply of the blood vessels. Okay, renal artery and the renal veins. Okay, and one more thing we should remember here, each kidney contain totally 2 kidneys, one pair of adrenal glands. One pair of adrenal glands above the kidneys. There are one pair of adrenal glands above the kidneys. Okay, this is all about the exterior structure or morphology of the kidneys. If you come to the interior structure, internal anatomy, so morphologically the kidney, length of the kidney of the human beings is almost all 11 to 13 centimeters, and the width is 5 to 7.5 centimeters, and weight is of about 150 to 250 grams. It is not at all fixed, so that 150 to 250 grams. Already we learned that outer surface convex, inner surface is concave. Okay, and inside that you can see the section of the kidney that divides the kidney into two regions. We can distinguish two regions, outer cortex and the inner medulla. So the outer cortex is somewhat thick red in color, and it is a granular part where it contains, in the cortex we will see the Malphigian bodies, Malphigian corpuscles, and the PCT, proximal convoluted tubule, DCT, distal convoluted tubule of the nephrons. They are present in the cortex. Whereas if you observe the see here, this is the entire kidney section, externally covered by the fibrous capsule, and internally we have divided it into the, so this is the cortical region where it contains Malphigian corpuscles and the PCT and DCT of the nephron, proximal convoluted tubule and the distal convoluted tubule of the nephron. Okay, whereas if you observe the light red colored agranular region is known as the medulla. Light red and agranular region of the inner side of the kidney is known as the medulla. Okay, and it consists of pyramid-like structures which are known as the renal pyramids. They are of about 8 to 15. 8 to 15 renal pyramids are there. 8 to 15 renal pyramids are there. Okay. These renal pyramids, for example, if you observe here, the ureter which enter into the kidney through the hilus, okay, it is formed into a funnel shaped structure. Singularly, it is called as a calyx. Plural is the calyces. Okay, this calyx, major calyx or the minor calyx, okay, they at the end they mixed up with the renal pyramids. Okay, this is the calyx and this is the renal pyramid. At the junction of both of these two, we will find out the renal papilla. We will find out the renal papilla. Right? So here you should remember, this is a ureter. It forms the pelvis. This pelvis get divided into funnel shaped structures. These structures are called as a calyx or calyces. Okay. So at the end of the calyces we have the renal papilla, through which actually get attached with the renal pyramids. There are 8 to 15 renal pyramids. Cone shaped renal pyramids are present in each and every kidney. They attaches with the calyx. Okay, and if you observe furthermore, here this cortex penetrates into the gaps of the calyces. This known as the columns of bertini. Columns of bertini. Okay, this is entire, it is the gaps in between the renal pyramids, which is known as the columns of bertini. Right? So this is all about the internal structure of the kidney. Internal structure of the kidney. And these renal pyramids attached with the calyces, and that enters into the pelvis, that opens into the renal ureters, and they attach with the urinary bladder. This is all about the anatomy of the kidney. Then come to the structure of the nephron. It's very, very important. Without the knowledge of structure of nephron, we may not understand the urine formation, so that we have to observe it very clearly. The nephrons of about each kidney, there are one pair of kidneys. Each kidney possesses 1.2 millions of nephrons. 1.2 millions of nephrons per kidney. So total 2 kidneys, 2.4 millions. 1 million means 10 lakhs. Okay, 24 lakhs of nephrons are present. 24 lakhs of nephrons. Each kidney is composed of numerous minute nephrons and ducts. Okay, these are also known as the uriniferous tubules. Nephrons are also known as the uriniferous tubules. Right? Nephrons are the structural and functional units of the kidney. Nephrons are the structural and functional units of the kidney. They are embedded in loose connective tissue. They are followed by the collecting system. In addition, kidneys contain a network of capillaries, lymph sinuses, nerves, and the interstitial fluid in the intercellular spaces. Intercellular spaces. Right? This is the introduction to nephrons. See here, if you see the section of the nephron, so this is small piece from the kidney, and here it is a elaborated structure of the nephrons with the blood capillaries, and here without blood capillaries. So this is the nephron. This is the nephron. Okay, let us discuss the structural components of the nephrons, structural components of the nephrons. Each nephron, for our convenience, get divided into two portions. One is known as the Malphigian body or renal corpuscle. Second one is renal tubule. Malphigian body and renal tubule. Malphigian body comprises two portions again, Bowman's capsule and the glomerulus, whereas the renal tubule it made up of proximal convoluted tubule (PCT), loop of Henle, and distal convoluted tubule. These are the parts of the nephron. Let us see what are they. See here, this portion is known as the Malphigian capsule or renal corpuscle, and remaining portion is known as the renal tubule up to this renal tubule. So now here in the Malphigian body there are two portions again. There are two portions again. In the Malphigian body we will see Bowman's capsule and the glomerulus. Here we will see, here there is a thin-walled cup-shaped structure. Thin-walled cup-shaped structure which is called as the Bowman's capsule. Right? Malphigian body means Bowman's capsule and the glomerulus. So this is the Bowman's capsule. This is the bowl-like structure is the Bowman's capsule. Right? Actually the Malphigian body begins as a blind tube in the outer portion of the cortex and consists of two parts, namely Bowman's capsule and the glomerulus. So this Bowman's capsule is a thin-walled, double-layered, cup-like structure. Thin-walled, double-layered, cup-like structure. Actually it is formed from the invagination of the tubule at the proximal end. Invagination of the tubule that makes the formation of the Bowman's capsule at the proximal end of the nephron, and it is consisting of simple squamous epithelial cells which are known as the podocytes. Very important in the examination point of view, and also in order to understand the ultrafiltration at the Malphigian body. It is thin-walled, double-layered, cup-shaped structure which is formed due to the invagination of the renal tube at the proximal end. Okay, it is made up of simple squamous epithelium cells which are known as the podocytes. Then if you observe the glomerulus is nothing but the network of renal artery simply. This is the Bowman's capsule and this is the glomerulus. So here if you observe, the renal artery enters into the kidney. One branch of renal artery which supplies the blood to the nephron is known as the afferent arteriole. This afferent arteriole enters into the lumen of the Bowman's capsule and it is get divided into small capillaries. For example, this is the afferent renal artery and it is divided into small capillaries, and all these capillaries again fuse to form the another vessel which is known as the efferent arteriole. Afferent arteriole enters into the Bowman's capsule and that will be get divided into branches, and all these branches again fuse to form another vessel which comes out as the efferent arteriole. So this capillary network which is formed by the combination of afferent arteriole and efferent arteriole is known as the glomerulus. Glomerulus is the capillary network which is formed in the lumen of the Bowman's capsule by the fusion of afferent arteriole and the efferent arteriole. One more thing here we have to remember, the size of the efferent arteriole comparatively less. The diameter of the efferent arteriole is comparatively less than the afferent arteriole. The size of the efferent arteriole is comparatively less than the afferent, renal afferent arteriole. Okay, so due to the combination of the afferent arterial branches and efferent arterial branches, they fuse to form a network-like structure, capillary network, that is nothing but the glomerulus. Both Bowman's capsule and glomerulus, both of them, they form the Malphigian body, that is the first part of the nephron. First part of the nephron. Okay, and here there is a importance in the urine formation of this Malphigian body. The wall of Bowman's capsule and walls of capillaries of glomerulus, they are formed by the single layer, that is endothelium. Glomerulus, that is made up of endothelium. They are in close contact. Bowman's capsule and the glomerulus, they are in close contact. So the blood in glomerular capillaries and the fluid in the lumen of the Bowman's capsule are separated by very thin barrier, that is nothing but the squamous epithelium of the Bowman's capsule and the endothelium of the blood capillaries of glomerulus, that makes the ultrafiltration of the urine, ultrafiltration of the blood. So that plays an important role in the urine formation, we have to remember. This is all about the first portion, that is the Malphigian body. Then coming to the renal tubule. In the renal tubule, we have three portions: proximal convoluted tubule, Henle's loop, and the distal convoluted tubule. Let us see what are they. Here, this is the neck region after the Bowman's capsule, otherwise a Malphigian body. From this region to this region, it is a proximal convoluted tubule, and this region to this region, it is the Henle's loop, and this region to this region, this is the distal convoluted tubule. Proximal convoluted tubule, loop of Henle, okay, and the distal convoluted tubule. Proximal convoluted tubule, Henle's loop, and the distal convoluted tubule. Let us see one by one what are the specialties of the renal tubule. Renal tubule, actually it is a narrow, delicate tubule arises from the posterior part of the Bowman's capsule, which is called as the neck region. It opens into a long, narrow convoluted tubule, which is divisible into three parts, namely proximal convoluted tubule, Henle's loop, and the distal convoluted tubule that enters into the collecting duct. See about the proximal convoluted tubule. The proximal convoluted tubule, the neck of Bowman's capsule continued as the proximal convoluted tubule. It is very long, convoluted, okay, which is commonly called as the PCT, proximal convoluted tubule. So here the important point to be remembered is it is lined by simple cuboidal epithelium, which has the brush border, is the specialty of the PCT. It is lined by simple cuboidal epithelium with the brush border. So what is the use of the brush border? It increases the area of absorption at the free surface. It increases the area of absorption. Why? Because without knowing the lining of the proximal convoluted tubule, we may not understand why there is a more and more selective reabsorption in the proximal convoluted tubule. Of about 65% of the ultrafiltrate get reabsorbed. So that we have to clearly understand what are the things which are kept inside. Okay. So here simple cuboidal epithelium tissue is there, which forms the brush border, which increases the area of absorption. Okay, already we discussed that the Malphigian body, proximal convoluted tubule, distal convoluted tubule, all of them, they are present in the cortical region. So below that, this is the medullary region. Okay, this is all about the proximal convoluted tubule. And coming to the loop of Henle. So here the proximal convoluted tubule leads into a hairpin-like tubule. Here it is a hairpin-like tubule which is known as the loop of Henle. Loop of Henle. It lies deep into the medulla. It present in the medullary region. And here it contains, so this region is the loop of Henle. Here it is a descending limb and here it is a ascending limb. Descending limb and the ascending limb. The walls of descending limb and the lower end of the ascending limb. Walls of descending limb and the lower end of the ascending limb. Okay, they are very thin and they are lined by simple squamous epithelium. Simple squamous epithelium. The distal portion of the ascending limb that is somewhat thick, and it is lined by simple cuboidal epithelium. Simple cuboidal epithelium. Descending limb and the anterior portion of the ascending limb, both of them, they are covered by the simple squamous epithelium, and the distal portion of the ascending limb somewhat thick and made up of, lined by simple cuboidal epithelium tissue. Right? Then coming to the distal convoluted tubule. Distal convoluted tubule. So this region, distal convoluted tubule. The ascending segment of loop of Henle enters into the cortex and becomes the distal convoluted tubule which lies near the Malphigian body. The lumen of early distal convoluted tubule is lined by simple cuboidal epithelium. The early, so it is a primary part. The lumen of early distal convoluted tubule is lined by the simple cuboidal epithelium, and the late distal convoluted tubule is lined by two types of cuboidal cells. This region, two types of cuboidal cells. The first one are the principal cells, and second kind of are the intercalated cells. Principal cells and intercalated cells. Okay, then intercalated cells are always with the microvilli. That is the specialty of the intercalated cells. The terminal part of the distal convoluted tubule is the collecting tubule. The distal part of the distal convoluted tubule is the collecting tubule. It opens into the collecting duct. Okay. Generally, distal convoluted tubule makes contact with the afferent arteriole. Okay. The cells of tubule are crowded in that region. They are known as a macula densa. The DCT interact with the afferent arteriole, and here there is a enrichment of cells, crowded cells. They are known as a macula densa. Along the side of the macula densa, the wall of the afferent arteriole continues the modified smooth muscle fibers. They are called as the juxtaglomerular cells, JG cells. Juxtaglomerular cells. Both the macula densa and the juxtaglomerular cells, they form the juxtaglomerular apparatus. Both the macula densa and the juxtaglomerular cells, both of them, they togetherly form JG cells, and the macula densa, they fuse to form the juxtaglomerular apparatus. So followed by, there is a collecting system. So this collecting tubule enters into the collecting duct. All the collecting ducts, they open into a ductus of Bellini. Right? Several collecting tubules open into the collecting duct. Collecting duct descend into the renal medulla again. Collecting duct also contain principal cells and the intercalated cells. In the renal medulla, these collecting ducts merge to form the duct of Bellini. It is also important. Duct of Bellini. The all of them, they open into the duct of Bellini. This duct of Bellini opens into the end of the renal papilla. Renal papilla open into the calyx. Calyx open into the pelvis, and pelvis open into the ureter. A ureter goes into the urinary bladder. Urinary bladder. So this is all about the renal tubule. This is all about the renal tubule. And coming to the capillary network of the nephron. So here we will see capillary network of the nephron. This is the afferent arteriole, and these are the efferent arterioles. These efferent arterioles, they spread over the proximal convoluted tubule and distal convoluted tubule, as we are observing in this figure. The efferent arterioles, they spread over the proximal convoluted tubule and the distal convoluted tubule. These are known as the peritubular network, peritubular capillaries. These are known as the peritubular capillaries or peritubular capillary network. And those arterioles which are present near the Henle loop, they are known as the vasa recta. This vasa recta has a functional efficiency or functional role in the counter current mechanism. So that we have to remember, the afferent arteriole get distributed over the proximal convoluted tubule and the distal convoluted tubule. This is known as the peritubular capillary network. And the same capillary network is also extended in the medulla near the Henle loop, that is known as the vasa recta. So all the peritubular capillary network and the vasa recta, finally they open into the small renal venules. These renal venules enter into the renal vein. This renal vein finally enters into mix up with the posterior vena cava, which collects impure blood from all the bottom parts of the body, behind parts of the body. So this is the capillary network of the nephron, capillary network of the nephron. Okay, and if you observe the portions of the kidney, here you can see this ureter, all the collecting ducts of Bellini, they enter into the renal papilla. Renal papilla enter into the calyx. Calyx open into the pelvis. Pelvis open into the ureter. Ureters are the small ducts, narrow ducts, which are arise from the both the kidney. They open into a sac-like structure which deposits urine temporarily. Ureters, that is, the pelvis of each kidney is continued as a ureter and emerges out at the hilus. Ureter is a long and muscular tube. Ureters of both sides extended posteriorly and open into the urinary bladder. What is a urinary bladder? Urinary bladder is a thin-walled, pear-shaped structure. Thin-walled, pear-shaped structure. It is a white transparent sac situated in the pelvic region, ventral to the rectum. What is the function actually? It temporarily stores the urine. Urinary bladder opens into the urinogenital canal by an opening, and it is surrounded by a internal sphincter. So it regulates the opening and closing of the urinary bladder, right? And the urine get excreted out, urinated through. There is a urinogenital aperture. Urinogenital aperture. Okay, so this is about the structure of the nephron, and the specialties we will see. The length of the nephron may be 20 to 45 mm, means 2 to 4.5 cm centimeters, 2 to 4.5 cm. And see the diameter of the proximal convoluted tubule, it is 0.06 mm. Diameter of loop of Henle, it is 0.02 mm. Diameter of collecting tubule, it is 0.2 mm. 0.2 mm. So already we discussed that 1.2 millions of nephrons per kidney, 2.4 millions for two kidneys. So if you arrange them side by side, then it is of about 120 kilometers in length. The total length of nephrons is of about 120 kilometers in length. So these are the special things about the nephron, right? Then furthermore, if you observe, there are two kinds of nephrons. Some of them are very small and confined to the cortical region, which are known as the cortical nephrons, and they contribute 80% of nephrons to the kidney. And another type of nephrons are the juxtamedullary nephrons. They comprise of about 20%. Cortical nephrons and the juxtamedullary nephrons. Let us see how they are looking like. Here we see, so this is the renal artery and renal vein. Okay, so these are the small nephrons. So they are glomerulus, okay, proximal convoluted, except the Henle's loop. The entire it is present in the cortical region only. So that they are the cortical nephrons, very small, but they contribute 80% of the nephrons of the kidney. Whereas the right side you see, so there are some large nephrons which are extended from cortical region into the deep medullary region. And one more difference in between these two cortical and juxtamedullary nephrons: in the cortical nephrons you may not see the peritubular capillary network and the vasa recta. Peritubular capillary network and the vasa recta are not seen, but where in the case of juxtamedullary nephrons we will see enrichment of peritubular capillary network and the vasa recta. Okay, and there is a functional difference in between. They are alternatively doing their work. Whenever the water amount, if the availability of the water is more, then the cortical nephrons, they actively participate in the excretion and they form the highly dilute urine. Whereas if there is a scarcity of the water and the excretion taken care by the juxtamedullary nephrons, okay, and they form hyperconcentrated urine, hypertonic urine, otherwise hyperconcentrated urine, and thereby, so these cortical nephrons, they form the dilute urine, and juxtamedullary nephrons, they form the hyperconcentrated urine, and thereby they regulate the water balance of the blood, balance of the osmosis of the osmotic nature of the blood. This is about the types of nephrons. And at last, see the functions of nephron. First and foremost function of the nephrons is the excretion. No doubt in this. So excretion of urea, uric acid, creatinine, ammonia, hippuric acid, different kinds of salts, and the bile pigments. Urea, uric acid, creatinine, ammonia, hippuric acid, salts, and the bile pigments. And second more function is regulation of homeostasis. How? By the regulation of water level in the body, by producing hypotonic and hypertonic urine, just now we discussed, and regulation of pH of blood, regulation of hydrogen ion concentration of blood, pH of blood. These are the basic functions of the kidney. If you see furthermore, so that will be discussed later on. Just let us see. Just remember, the function of glomerulus is the ultrafiltration. The next class we will see this. Bowman's capsule receives the glomerular filtrate. The proximal convoluted tubule, it reabsorbs the most of the water, glucose, amino acids, inorganic salts of the sodium, potassium, and chlorides. And the function of the loop of Henle is reabsorption of water and sodium ions. And the function of distal convoluted tubule is the tubular secretion of potassium and the hydrogen ions. Chemicals like drugs, antibiotics, etc., and absorption of small amount of water and ions. So of about 9% of water and ions. And finally, the function of collecting duct is reabsorption of water of about 10% and collect and transport the leftover filtrate that is in the form of urine. So these are the functions performed by the nephron, different parts of nephron. So until this we have covered the structure of kidney, right, and the structure of nephron and their parts and their functions. Thank you one and all.